Welcome to our series of podcasts on lab techniques. This guide will aid biomedical science students in their learning of the practical and theoretical aspects of tissue culture, with the hope that students will gain insight and skills that can be applied to their lab work in the future. In this podcast, we'll be explaining the themes around tissue culture. We'll explore the history of tissue culture, the type of cells used in tissue culture, equipment, substrates and controls required for tissue culture, and application of tissue culture. This podcast will also contain videos of aseptic technique and the procedure of cell culture. What is tissue culture? In simple terms, it is a technique where cells of interest, for example fibroblasts, are grown in vitro outside the body or organism, under specific conditions which allow the cells to grow and divide. Tissue culture is sometimes referred to as cell culture. The history of tissue culture originates from the beginning of the 20th century, when the first cell culture experiment was carried out. Ross Harrison adapted the hanging drop technique to successfully carry out the first cell culture experiment. Harrison cultured nerve fibers from embryonic frog neuronal tubes using his adapted hanging drop technique. Harrison's technique became a major tool until the 1950s for other researchers developing cell culture techniques and cell lines. Dr. George Gay aided the development of tissue culture in a number of ways. Development of the roller tube technique saw the explants being placed in a plasma-coated test tube, which was then placed in a roller machine and rotated as seen here. This technique was useful in the growth of viruses in cultured cells. From the 1930s onwards, scientists would use this technique for the growth of viruses and possible vaccinations. The most significant contribution to the history of cell culture from Gay was his work on HeLa cells. In 1951, Henrietta Lacks died of an aggressive adenocarcinoma of the cervix. With the use of his roller tube technique and trypsin, Dr. Gay dissected a small part of this tumour and cultured the cells to produce the first human cell line, commonly known as a HeLa cell line. The development of the polio vaccine was based on the work of Enders, Weller and Robin, who won a Nobel Prize in 1954 for their discovery of the growth of the polio virus in 1949. Jonas Salk used the knowledge of the growth of the polio virus and the aid of Media 199 to culture monkey kidney cells containing the polio virus. Salk developed the first effective inactivated polio vaccine in 1952. Aseptic technique is a procedure used when working with cells. The technique aims to reduce the risk of contamination. Contaminants such as mycoplasma, yeast, bacterial and fungal spores need to be controlled and kept to a minimum. A single incident of contamination can spread throughout a lab and have the potential to shut a whole lab down. Equipment such as pipettes and tissue culture flasks can be purchased in a sterilised packaging from the manufacturer. Equipment is only opened when placed in a laminar hood, reducing the risk of exposure to contaminants. Laboratory gloves should be used throughout your experiment as it prevents the spread of contamination from exposed areas such as work surfaces and the handle themselves to sterile surfaces. 70% ethanol is widely used alcohol in labs. It is used for sterilisation and is used to remove contaminants found on work surfaces, solution bottles and the laminar hood. A key piece of equipment needed for aseptic technique is a laminar hood. This provides an aseptic work area by protecting the area and cells from contaminants. The laminar hood allows the maintenance of a constant and stable flow of air, which simply means filtered air can flow in multiple directions, horizontally and vertically. The important point to remember is that the vertical airflow shown in the picture flows from the top of the hood onto the work surface and recirculates, providing protection to the user and the cells. Before working in a laminar hood, we need to ensure the glass of the hood is sterile. As you can see, the glass is worked down using 70% ethanol. The surfaces inside the hood are also worked down with 70% ethanol to ensure none of the equipment or the cell culture comes into contact with contaminants. Finally, sterilising the materials that you will use, such as media bottles, is recommended, as there is a risk that they may have come into contact with contaminants during storage in the fridge. For cells to grow at their optimal rate, a controlled environment is required. Factors such as carbon dioxide levels, temperature and humidity are monitored. A pH between 7.2 to 7.4, a temperature of 37 degrees Celsius, a high humidity of 95%, and carbon dioxide levels of 5% are usually required for optimal growth. Filtered air is recirculated through the incubator to minimise contamination. Copper sulfate crystals can be used at the bottom of the chamber and are responsible for preventing mould which may form due to the high humidity. Media is essential for the growth of cells. Cells are placed in the media, which helps to control the osmotic pressure and the pH of the culture. Each media differs in its composition of amino acids, sodium bicarbonate buffers, and pH. The two media shown here are commonly used in the lab, but there is a large range of media that vary depending on the requirements of the cultured cells. RPMI 1640 can be used for the growth of mammalian cells such as leukocytes and carcinoma cells. It contains a glutathione, a reducing agent, and a high concentration of vitamin B12. DMEM F12, Delbecco's modified Eagle medium nutrition mixture F12, can be used for the growth of mammalian cells such as rat fibroblasts. It contains a high concentration of glucose, amino acids, and vitamins. Most media used in labs contain a sodium buffer system, which helps to maintain carbon dioxide and pH. 10% fetal bovine serum may also be found in media, as it supplies proteins, lipids, and growth factors. When using media, be sure to check the expiration dates of your solution, as it can affect your experiment if not in date. There are two types of cell culture techniques: mechanical disaggregation and enzymatic disaggregation. Mechanical disaggregation involves dissociation of cells in a culture by forcing the cells through a series of sieves. This technique is rarely used in labs. Enzymatic disaggregation involves the dissociation of cultured cells using enzymes. This technique is commonly used in labs and will be described in detail later in the podcast. Adherent cells are cells in culture that need to be attached to a substrate in order to grow and divide further. Examples of adherent cells include neural cells and liver cells. Adherent cells are dissociated by enzymatic disaggregation in order to allow a cell population to continue to grow and divide. This is known as passaging the cells. Suspension cells are already in suspension and do not need to be dissociated from a substrate in order to passage the cells. However, the media will become exhausted of nutrients during cell growth and will need to be replenished. This can be done by removal of old media and addition of fresh media. Examples of these cells include erythrocytes and osteoblasts. Adherent cells have been incubated with media for 2 days, allowing the cells to grow and divide. The cells are grown in flasks varying in sizes. The one shown here is known as a medium-sized T75 because it has 75 square centimeters of area for the cells to grow upon. Smaller and larger sized flasks are commonly used. The size chosen by a researcher will be dependent on the number of cells required for their experiment. Once taken out of the incubator, the cells are examined under a microscope to make sure they are alive and healthy. We also need to identify how confluent the cells are. This term refers to the number of cells in the flask and if they have room to grow. As you can see from this picture, the cells are 100% confluent. This means the cells no longer have room to grow, as all of the surface area is occupied by the cells, and the researcher will need to passage. This is an example of cells that are around 50 to 60% confluent, which means approximately half the area of the flask is occupied by the cells, but there is still space for them to continue to grow and divide. To passage the cells, the media bathing the cells first needs to be removed. This can be done using a glass pipette attached to a suction tube as shown here. It is recommended that you ensure the suction tube is sterile before use to prevent contamination of your culture. Don't touch the bottom of the flask as this can disturb the cells. Tilt the flask instead, as this ensures that all the media is in one corner and will make it easier to aspirate. Once all of the media has been removed from the flask, PBS, a buffered saline solution, is added to the cells. PBS is added using a pipette. Note that the pipette is opened under the laminar hood to ensure it remains sterile. For this size of flask, around 10 milliliters of PBS at room temperature is added in order to wash the cells and remove media and the serum. To ensure full coverage of the cells with PBS, the flask is swirled around to ensure all cells are washed. The PBS is then aspirated out. The PBS wash step is required as residual serum can inhibit enzymatic disaggregation of the cells. Once the cells have been washed with PBS, they can be dissociated from the flask. This can be done using proteolytic enzymes such as trypsin, which disrupts both cell-cell interactions and cell-flask interactions. In this video, the researcher is using a non-animal based alternative called Triple Express. 5 ml of Triple Express is pre-warmed to 37 degrees Celsius in a water bath. It is pipetted into the flask. The flask is then swirled to ensure full coverage and placed in the incubator for 3 to 5 minutes. After incubation, the researcher will check the cells under the microscope to check whether the cells have dissociated from one another. Dissociated cells will look circular in appearance, and in this example, approximately 90% of the cells were observed to be detached. If cells still remain attached to the flask at this point, the flask can be given a gentle tap on the side, and this often dislodges the cells. Certain cell types can be more difficult to dissociate than others, and may need longer incubation at 37 degrees Celsius. If using trypsin, it is important that cells are incubated in this solution for as short a time as necessary, as prolonged exposure to trypsin can damage the cells. To collect all of the cells in solution, tilt the flask to ensure that the cells collect in one corner. Once the adherent cells are in solution, they can be pipetted out of the flask into a sterile tube. To inactivate the triple express, an equal volume of media can simply be added to the cell solution to dilute the enzyme. If using trypsin, it will be important to add media containing serum in order to inactivate the enzyme. The tube is centrifuged at 100 G to form a cell pellet at the bottom of the tube. Most centrifuges only have settings for speed in revolutions per minute or RPM, so the RPM setting you use will be dependent upon the centrifuge model and the rotor used. The triple express media solution is removed at this point, leaving the cell pellet at the bottom of the tube. One tip to aid cell suspension is to flip the bottom of the tube a few times before resuspending the cells in fresh media. This tends to help avoid cell clumping. At this point, the cells can be reseeded or passaged into sterile culture flasks at different ratios. Ratios can vary from 1 to 3 to 1 to 20, depending on the growth rate of the cells in culture, the size and number of flasks, and the experimental requirements of the researcher. We now have an understanding of how cells are cultured, but what are cultured cells used for? Culture cells are widely used in the pharmaceutical industry in order to develop new drugs and to investigate the therapeutic effects and potential side effects of drugs. Reconstructive surgery using skin grafts is an area of medicine that requires cultured cells. Skin grafts can be used to repair skin that has been badly burned or injured. This procedure involves the removal of tissue, in this case skin, from an unaffected area. The cells from the skin are then grown and transplanted to the area that has been affected. Cell culture is used to produce in vitro human models of the brain in neuroscience. Researchers use brain cell cultures to study aspects of the human brain. In vitro models are used in the understanding of brain signaling pathways, the normal brain function, and pathological processes involved in neurodegeneration. We hope that you have now gained some insight into the theoretical and practical aspects of tissue culture. In the hope by the end of this podcast, you'll be able to define the term tissue culture, understand the importance of aseptic technique, have an appreciation for the practical aspects of cell culture techniques, and the use of cultured cells in science and medicine.